Hello, everyone. My name is Olivia Sourwine, and I had the privilege of working with Dr. Choi as my mentor in the Human Auditory and Neuroscience Lab. And the title of our project is How Does the Brain Predict Who's Speaking? So one of the most common complaints of individuals with hearing loss is how difficult it is for them to listen and understand their conversation partner in background noise. They often report high levels of strain and fatigue, and past research supports that it is easier to listen to a familiar voice in background noise than an unfamiliar voice. They found stronger speech-evoked brain activity in the familiar voice. Therefore, the purpose of our experiment is to investigate neural substrates of speaker identity tracking during speech and noise. And although we do hope to make clinical advancements in individuals with hearing loss, we first will investigate the typical neuromechanisms in normal hearing participants. The predictive coding theory believes that the brain generates predictions of what events will occur in its sensory environments based on its internal representations of the world. Later, it will sample the sensory inputs and update its predictions. There is physiological evidence that we use neurocognitive processing, and this can be investigated through EEG by looking into different brain oscillations. In this particular study, we will be focusing on gamma and beta oscillations. Gamma oscillations have a frequency of 30 Hz and above, and is dominated by sensory sampling. However, beta is about 15 Hz and is dominated by top-down processing. Additionally, we can look into where the brain waves are coming from in the brain. The supra-temporal gyrus in the yellow color is a sensory area, and the inferior frontal gyrus in blue is a higher level speech and language area that relies on top-down processing. In order to understand speaker identity processing in noisy environments, we do rely on the frequency range of the cortical oscillations and also their location in the brain. So our hypotheses are that beta oscillations will be located in the inferior frontal gyrus for the speaker identity cue, and the gamma oscillation will be present in the supratemporal gyrus for the non-speaker identity cue. And then to the left, you can see the trial structure. The cued condition had a single voice that matched the target word in the background noise, and then the non-cued condition had two speakers in which you couldn't predict the voice that was going to be spoken later on. And then now we will go over the EEG results. On the left, we have three different spectrograms with time on the x-axis, frequency on the y-axis, and amplitude through the color shading. During the preparatory period, which is where the speaker identity predicting or tracking would take place, we do see in that top cued spectrogram at 0.5 seconds, there is a strong intensity at 15 Hz, which indicates a beta oscillation. However, there is no gamma oscillation between 37 and 45 Hz. For the second panel, the no-Q spectrogram, we do see the gamma oscillation at 0 seconds. However, there is no beta oscillation at 15 Hz. Additionally, the third panel measures the t-scores, which are used to measure the difference in the effects of the conditions. The red color is positive values and means that the cued condition had significant and greater effects than the non-cued condition. And then the blue color is negative and represents that the non-cued condition had stronger effects than the cued condition. And then the neutral green color surrounding the blue and the red colors indicates that the p-values were greater than 0.05 and there is no significant difference. Therefore, in the last panel, we do see gamma was significantly larger for the non-cued condition and beta was significantly larger for the cued condition. And then while we are investigating the brain regions at the very bottom of the slide, we do see gamma was located in the supratemporal gyrus in both hemispheres. However, beta was located in only the left hemisphere of the inferior frontal gyrus. In conclusion, our results do support the predictive coding theory. In noisy environments, it uses top-down processing indicated through beta waves in speech and language processing areas when predicting and using that speaker identity voice. However, without that speaker identity, the brain uses bottom-up processing and sensory sampling and indicated through gamma waves in sensory areas of the brain. This is because it needs to be open for all voices. These results did support our hypotheses. In the future, we do hope to increase the sample size and hope to test individuals with hearing loss to make clinical advancements and to mitigate communication barriers. And then here's all of our references. Thank you so much for listening.